Greetings, parasite ecologists. Today, I'm here with Armand Kuris, who is a professor at the University of California in Santa Barbara. And at this point in the video series, we've covered so many different parasite taxa. Protozoan, metazoan, we've talked about a couple of viruses and bacteria, and we're here today to ask, who cares? Why did we make an entire video series about parasites? Armand is here to help us answer that question. Armand, thank you for joining us.
It's my pleasure, Chelsea. Glad to be here.
Today, I want to talk about your paper from 2008, the one titled Ecosystem Energetic Implications of Parasite and Free-Living Biomass in 3 Estuaries. And I want to start just by finding out what inspired you to write this paper.
The inspiration was the broad issue of what is the role of infectious diseases in the world at large, at the ecosystem level. And we know if in ecology that predation is important and competition, resources, disturbances. Infectious diseases are hidden inside hosts. And so we sought to reveal that. And the way we felt we could get the first serious look at this was to figure out how much biomass the parasites was parasitic at the ecosystem level.
So why did you choose to focus on biomass?
Size is a surrogate for importance. At the individual level, it's obvious that what a single elephant does is more important than what a single mouse does. And so by looking at the biomass and accumulating, maybe all the mice actually have a bigger role than that one elephant, we will have the first scale look at the importance of infectious diseases by quantifying them inside their hosts.
Was biomass something that people thought about as a proxy for ecological importance before you used it as a proxy for the importance of parasites?
I think always that has been the first thing, because an actual study quantifying energy flow is very difficult and has never been attempted at an ecosystem level.
Were there any food webs before your work that had incorporated parasites into them?
There were a few that put some parasites into the food web. But just a selection in a few hosts and only of a few kinds of parasites. And what we went for was, let's treat every host species in the ecosystem with equal care about their abundance and about their biomass, and then again, with a careful sampling program, attempt to quantify all the parasites we could identify and quantify. So that's what we sought out to do. And we did it in these estuaries because we thought that they were tractable ecosystems.
Okay, so tell me more about these 3 estuaries where you worked. Why specifically did you choose these places?
Well, we're based in Santa Barbara, California, so we have a very nice estuary just 15 miles south of campus, Carpinteria Salt Marsh. And we had done work there on related projects. And to make it sure that we were talking not about one marsh, but about the ecosystem of estuaries, we took 2 other marshes that had not been heavily impacted by human activity, Estero de Punta Banda, just south of the city of Ensenada in Mexico, and Bahia San Quintin, which is in Baja California, about a 5-hour drive south of the U.S.-Mexico border.
And you chose those ecosystems because you thought that they were relatively simple and had few free-living species and therefore were tractable to quantify, both in terms of their free-living biomass and their parasitic biomass.
Yeah. So in total, there were about 200 species of free-living organisms that were larger than about 1 millimeter in length. And we were able to identify over 160 parasitic species among those.
So the idea basically is to look at the entire food web, all the free-living and parasitic species, and quantify the biomass of everyone involved in that food web. But that involves looking at things that are living in the sediment, like polychaetes, and things that are on top of the sediment, like snails, and then also things that are flying through the marsh, like birds. How did you handle all those different groups of organisms?
So it was obvious, like from what you're implying, is that doing one sampling program would sample some of those really well and some of those terribly. So we wanted the best estimate of the density of all of the hosts, and then an appropriate, again, fair sampling of each of those types of hosts. So for fishes, we used barrier nets and caught all the fishes in a channel of the estuary. For the snails, we had quadrats on the surface of the mud. For clams, we did cores. So we used an appropriate sampling method for each of the major categories of creatures.
And then for each of these marshes, you calculated the biomass density for all of the species that you targeted. Why'd you use biomass density?
The density was based on area and abundance of these things. You have to make it per unit area.
Why?
Because the number of snails to match up with the number of fishes, they're not even in the same part of the habitat in the estuary, and that would also let us compare the different estuaries. The two estuaries in Mexico were, for instance, a fair bit bigger than the Carpentria Salt Marsh estuary. So we had to make it per unit area. And again, we did that for all of the species.
So you wanted to make sure you were comparing apples to apples.
Exactly.
Yeah, okay. This sounds like a gargantuan field effort. What did it actually look like for the team to be doing this kind of sampling?
Very muddy. It required a team effort where the direction came from the principal investigators, Kevin Lafferty and myself. And then on down, there were captains for working with the fishes and the birds and the invertebrates. And all the way on down to a technical person saying, here, lay out this transect line over here. And then the senior people, Kevin and I and some other people, would actually then reinsert ourselves on the bottom and pick up the snails along with undergraduate students, graduate students, technicians, and everybody. So we actually had a feel for literally how the data was collected in addition to planning the overall design.
And then what about the parasites? How did you get at parasite biomass?
Then we again had a randomized sampling related to size of the animals for almost any species. Big animals have been out there longer than the small individuals of that species, have accumulated more parasites. So we had to pay also attention to the relative density of different sizes of these species, and then dissected them with everybody being trained up not only to recognize the parasites that we knew that they were there, but also keep your eyes open because we are now looking at species where nobody has ever dissected them for parasites. And our team had to be able to immediately recognize, this is something new. How can we identify it? What is it? And get the quantified data, which would mean the size of the animal and its species and its abundance in those.
Did you find new parasite species?
Absolutely.
Have they been described now?
Some of them have been described, yes.
So what would an average day in the field look like for your team?
You work at low tide. So tide has to be out. So depending on where you are, you could be going out, driving out to the place before dawn, or coming in at the late afternoon and coming in at night. And so then you go on out there, and the team is organized so that you know where you're going. Somebody's in charge of directing where the quadrats are, and then the various people are there to pick up the crabs. Meanwhile, there's a battery of microscopes back in some sort of lab facility where the dissectors are again trained to work with crab dissectors or snail dissectors or fish and be backup so that there's a dissector and a scribe who is quantifying how many and what you've got.
And while you were in the field, did you have any sense of what the data were ultimately going to tell you, or did you have to wait until the analytical stage before you saw what the data were going to teach you?
Well, to be completely honest, we thought there were a lot of parasites out there. So we were expecting to find things. I mean, on some of these species that were abundant, for instance, the most common crabs, the most common snails, the fishes, there had been work about what was in those things. So we knew we were going to find things. How much, we did not know, because we did not have a sense of the density of these things.
So you went into the project expecting that you were going to find a reasonable abundance of parasite biomass?
Who would do all that crazy work to find nothing?
Fair point. So tell us about what you did find. What did you learn?
So parasites are found throughout the system, and what we found, I think most importantly, when you made it by density, that some of the most abundant species in the marshes, which were, let's say, the two most abundant were the snails and the crabs, was that they had a very rich parasite fauna that accounted for, as much biomass as did the top predators that are birds. And in particular, in the snails and in the crabs at some of these locations, 1 or 2 or 3% of the tissues of the host was actually parasitic. 1% sounds like, well, that's not much. But actually, if you start thinking about yourself and you start thinking about what parts of the body are 1% of you, that's much more than your eyes or your kidneys or your pancreas or reproductive organs. And parasites are generally, they're not feeding off the fat, they're feeding off the good stuff. And so that being 1, 2, or 3% is a huge fraction of the host. And overall, as I said, the biomass in total in those groups was actually that of top predators. And ecologists all over the world are always acknowledging that, let's say the lions play an important role in the Serengeti ecosystem on the Great Plains of Africa. And wolves do in the north, and bears do. Top predators are very important. And by looking at biomass, this suggests that parasites may well play as important a role in ecosystems as does predation.
So previous folks have looked to food webs and to estimates of biomass in the various components of food webs as a way to understand the importance of particular taxa in an ecosystem. And they found, using that metric, that top predators are important both in this energetic term, but also in other ways by changing things in the food web. And you found that at least for these salt marsh ecosystems, the top predators, which are birds, are outweighed by the parasites. There's more biomass that's parasitic than there is biomass that's top predator or bird.
Predation is sort of wham, cheetah eats antelope, antelope's gone. Mm-hmm. And the next day or a few days later, it happens again. Parasitism, most of the parasites in our ecosystem actually are there for the life of the host after it's infected. So that's a little bit of a suck of energy from the host every day. And the most important snails in that marsh, which reach densities of 1,000 per square meter, live 20 to 40 years. infected most of them by the time they're about 4. So then you just start adding up that suck. That's just a lot of energy flowing through the ecosystem that is going to the parasite. Two other features that play a big role in this. Many of these parasites are parasitic castrators. And what that does is that means that the host lives on. It never makes snail babies. It now makes just worm babies, trematode babies. And so that dynamic means that this energy drain plays out in an enormous way in the success of the snails that are no longer snails, but they're actually competing with the real snails. And then the second thing is many of the trophically transmitted parasites, where they're in a prey host, like a killifish, are ultimately going to mature when something like a right host, like a bird, a fish-eating bird, eats the killifish. And they are powerful behavior modifiers. We've shown this for some of these. Many other studies have shown this is quite common for prey hosts to be modified, their behavior to be modified, making them more susceptible to being consumed by the final host, the predator host. So they're also directing, in a certain sense, I don't want to get too biblical here, but who shall live and who shall die each day by making some hosts much more likely to be consumed because their behavior has been modified to make them more accessible to the right predator host.
So in addition to composing a huge fraction of the biomass in these ecosystems, the parasites are also directing how energy flows through the food web.
Very much.
That's something that your study doesn't necessarily quantify, but seeing how much biomass there is in parasitism, you can just imagine how much control they have over those energy pathways.
Right. So our biomass study then links to lots of studies about behavior modification and also links back to the food web, because it's indicating why some of those strands in that food web are as strong as they are, because the parasites are moving things in a certain direction to a fair degree. Or, in the case of the castrators, you think that that snail is eating diatoms, but actually it's the worm with its snail mouth that is eating those diatoms.
You've sometimes called this the hand in the puppet, yes?
That's been called the hand in the puppet. I won't take credit for coining that phrase.
What about the division of biomass among different parasite groups? Who's composing the majority of parasite biomass?
So in these estuaries, and this is probably because trematodes play such a big role, the castrators actually on their own match up with the birds, the top predators. The trophically transmitted parasites are also a very big biomass component, almost as big. What we call typical parasites, the usual worm that lives in the gut of a host, actually do not have anywhere near the biomass component.
Why do you think that is?
I would describe, because especially when so many of the ones that are in the final host came to that host through predator host eating prey host, that the growth of the parasite was in the earlier larval stages. That's where the energy flow is. What happens in the final host for, let's say, the adult tapeworm is it's almost like the flowering stage of a plant. They reproduce, they live a short time. They don't do much damage to those final hosts because birds, by the way, are not stupid. If I can look over the bridge and see the killifish flashing at the surface caused by its parasites, and then the bird goes down and eats that, why is it going out of its way to eating a parasitized fish? It's going there because the parasite is going to do next to no damage in the bird, very little pathology. To use a phrase coined by Kevin Lafferty, the pizza boy delivers the pizza. How much do you tip the pizza boy? That's the cost from the parasite.
So you found in these 3 salt marshes that Parasites comprise a huge proportion of total biomass, rivaling that of top predators. And your study was the first of its kind in that it estimated this biomass across the entire food web. In the studies that have come since, has the same thing been true? Are parasites a big component of biomass no matter what ecosystem you look at, just in salt marshes? Or is there a rule about where you're going to find a lot of parasite biomass?
We're still working on that. That, to me, is the really big-scale unresolved question of what is the role of the infectious process in ecosystems. So to get to that, you have to look at very different ecosystems. We started with a tractable ecosystem. There are ecosystems that are hard to get to, the hydrothermal vents. Lauren Dykman has just done, just a couple of years ago, the first comprehensive study of parasites in those animals, the tube worms, et cetera, at the hydrothermal vents. Hard to get to. There are other ecosystems that are so diverse, coral reefs, rainforests. In fact, nobody has done this in a terrestrial ecosystem yet. Aquatic ecosystems have been more tractable. To pick one ecosystem where they've now done a lot of work, way up in northern Norway, there's a huge lake, Takvatn. And while our world, you could say, in the salt marshes are dominated by trematodes, it's a tapeworm world up there, where tapeworms accumulate through the food chain, ending up in these Arctic char and brown trout, fishes that are this long, and accumulate tapeworms there. And by the relative numbers are similar, and that's by numbers of species, a somewhat simpler ecosystem than ours. But so there is now some significant studies. Most recently, Zoe Ziltz has looked at the parasites in the rocky intertidal zone. So that's an ecosystem that is almost as rich as a coral reef. And so by no means could anybody examine all of the different invertebrates and the fishes and the birds that use that and the interface between the land and the sea. And so there, in a first really serious good look at things, Zoe has found that there's far fewer parasites in that ecosystem. It's quite an unknown issue as to why that is the case. But that's an example of you only get to figure that, to get a sense of those questions when you begin to find an ecosystem that isn't meeting the sort of quick thought of, more hosts mean more parasites.
Do you have any ideas about why Lake Takvatn in northern Norway is a tapeworm world and Carpentaria Salt Marsh is a trematode world?
So trematodes all need snails, and freshwater snails are smaller than the marine snails. They are less abundant in biomass, literally, than these snails are in Carpentaria, or the clams in Carpentaria, the mollusks there. So reducing mollusks and then getting them up into the food chain, they're there. There are trematodes there, quite a few species actually. However, their biomass is low because the snail hosts are small, so even though they're castrators, that's a high percentage of just a little smidge of tissue. And then they end up in the fish as a little tiny worm. Whereas the tapeworms are accumulating through the ecosystem. They're already pretty large, often a few centimeters, in the intermediate fish hosts, and then they accumulate in these large fishes. So that in many cases for fish that are game fish in some of these lakes that are natural with the birds that rain the tapeworm eggs down on the lake, you wouldn't want to eat those fishes because they're riddled with tapeworms throughout their musculature.
Just a little bit of extra protein, right?
Probably taste okay. There is a recipe in New Guinea for eating larval roundworms, but I mean...
Have you tried it?
No.
Would you try it?
I would try trematode.
But not roundworm?
I don't think so.
Why not?
I don't know. Ask my wife. She's more adventurous about food than I am.
So if there was one take-home message that you want us to take away from this paper, what would it be?
Don't forget about the parasites. They may be important players in almost any level of dynamic, whether you're concerned with the behavior of your host, the population abundance of the host, the health of the host, or in fact the functioning of an ecosystem. And if they're meddled with, you may or may not like the outcome.
All right. So today we've heard about how, at least in some circumstances, parasites can be really energetically important, composing a huge proportion of biomass in ecosystems. Yeah, they're small, sometimes they're hard to find, they're hidden inside of their hosts, and historically we've ignored them. But as Armand's research shows, ignoring them probably isn't the safest bet. So who cares about parasites? Hopefully, by this point in the video series, the answer is you. Armand, thank you so much for joining us.
Thank you, Chelsea. This was a lot of fun.
I'm glad. And we'll see you in the next video.